Hello and welcome to this IChemE Global Awards 2021 webinar. My name is Matt Stalker and I'm your host for today's presentations. The IChemE Global Awards celebrate chemical, process and biochemical engineering excellence and are widely considered to be the world's most prestigious chemical engineering awards. Today we'll be revealing the winner of the IChemE Biochemical Engineering Award sponsored by WSP. This award recognises the best project, process or product to demonstrate excellence in the application of biochemical engineering. But before we check out our contenders, let's hear from our sponsor. It's WSP's Technical Engineering Director, Simon Lewis.
Good morning, everyone. I hope everybody can hear me loud and clear. And I'd like to say, first of all, congratulations to all of our finalists for getting to this stage in the process. I thought I'd start with a few personal reflections of mine, really, as part of an introduction to this award. The IChemE Global Awards have become hugely significant in the global calendar within our profession and within the industries in which we work. It's really a great opportunity to showcase some of the fantastic work that we're doing across all of the sectors. And as I said, congratulations to all of our finalists for getting to this point. I think you should all be extremely proud of the projects that you're doing and the work that you're doing and the difference which some of these projects are now making to our society. It may feel to some of you that you've actually had to deliver this project twice now, firstly in delivering the project and secondly in producing the submission for the IChemE Global Awards, and it might be an extra piece of work that you had to do to do that. But you know, I'd like to thank everybody for taking the time to prepare the submission, you know, because it does give us an opportunity to properly showcase everything that we're doing across multiple sectors and multiple areas. And I hope that you know, being part of this sort of finalist presentation set, it'll make you feel a very important part of a collective in some of the work that we're doing. I had the privilege a few years ago to be part of the team that won the Global Team Award. It wasn't something that we thought we'd win. It was, you know, we were up against some very strong projects, even though we felt very proud of what we'd done in our project. But you know, certainly it felt very, very prestigious, and it felt great to be part of the whole IChemE Global Awards event. You know, and more than just the winning, really, it was very much about the team actually taking a bit of time out for ourselves and doing a bit of self-reflection on the project that we'd achieved, and giving ourselves a pat on the back and celebrating. And I think, you know, of course there will be a winner today, but I think for all of our finalists, what I'd sort of reflect and ask of you really is take the time to use this as a bit of self-reflection with you and your team, and have a look at exactly what you've achieved and congratulate yourselves for doing that. I think as engineers we're conditioned to deliver projects and be very modest about it, and then move on to the next project, and sometimes not. You know, we don't do enough of the congratulating ourselves and properly looking at exactly what it is we've achieved, which in many cases is very significant. I mentioned about the making a difference and delivering projects which make a difference, and you know, I think the world is obviously throwing a lot of challenges at us, and that will continue. And as engineers and scientists, I think we're the ones who are going to have to provide some of the solutions to some of the problems of the future, or certainly play a big part in providing the solutions. Obviously, there will be a political element to providing the solutions, but technically, you know, it's down to us. We're the people that can deliver, you know, the technical solutions to some of the issues that we're now facing. The slide that I put up there, it's actually some text from our web page, and it's one of the first things that you get to when you arrive at WSP's website, under the banner of what we stand for. And when I read it, every time I read it, it really sort of hits home very hard to me. We believe that for societies to thrive, we must hold ourselves accountable for tomorrow. And I think, as engineers, that's hugely important. We have a huge legacy that we're going to bestow on the world, and I think it's up to us to shape that and determine exactly what that legacy is. And certainly, it's something that is very close to my heart, and part of the reason I think I'm working for an organisation like WSP is because I feel that there is a sort of contribution and a value towards the sort of the future success of our society. And I guess that's one of the reasons why we sponsor awards such as the Biochemical Engineering Award, because we see that as very much part of the solution going forward. So yes, biochemical engineering, it's a part of the solution. We are engineering processes to create products which range from medicines through to renewable energy, low energy synthesis routes for established chemical manufacture, as well as greener solutions for things like waste management and waste treatment. And it's biochemical engineers that are responsible for tackling a lot of those sorts of issues, or certainly providing a solution which maybe complements other areas that are contributing to the overall global solution. Thinking about things like vaccines for against the pandemic, for example, and other viruses, flu, stem cell therapies to cure blindness, biofuels from algae, and the list goes on. This shows areas where WSP are supporting clients across the globe in pharmaceutical and biotechnology areas. So again, it shows the importance that we're putting to this particular sector. So we deliver projects across the globe, and from the UK, I'm very proud to say that we are our WSP Centre of Excellence for Process Engineering and Pharmaceutical Engineering is based in the UK, and it's a team that I'm very proud to be part of. But also we've got expertise, specialist expertise across other regions. So in Europe and in North America, for example, we have high containment specialists, and it's through those connections within the organisation that we can properly service clients across this sort of geographical stretch. Globally, we have around 55,000 people and 7,000 in the UK, and we support clients through all phases of the project lifecycle, from concept and master planning right the way through to delivery, execution, commissioning, and even through to decommissioning. So, and we do that through a very collaborative approach, both internally, finding the right expertise within our own organisation, but also collaborating with our clients and with the industry sectors to shape the right project. When you think about multidiscipline, it's more than just the traditional process industry multidiscipline of chemical engineering, mechanical, instrument control, electrical, etc. We also have reach into more specialist areas of things like utilities, highways engineering, environmental, building and structures. And that might seem a little bit sort of a tenuous link when we're delivering process industry projects, but I think when you consider the global scale of some of the things, some of the challenges which we have across the world, then it really is going to need some very clever thinking from many different specialisms and sectors. And there are a few examples there of some of the organisations we've worked with recently, and some sort of screenshots of some of the work that we've done. So just to conclude, really, a few areas where biochemical engineering is really impacting on the future, and where the application is really going to provide us with the sort of solutions that we need. And I've just picked three as just to talk about for a few moments: food production, energy, and pharmaceutical development. So when we think about food production, then the challenge for us really is how do we feed our ever-increasing global population, and it is it's a way we need to use our resources much more efficiently, find lower intensity ways of producing food. So for example, alternative food sources, synthetic proteins, etc. Novel food production to reduce the amount of energy that we're using, and thinking about things like packaging and sustainable packaging that using sort of biodegradable and compostable food packaging produced from sort of wood-based products, cellulose fibers, etc., etc. So food production, it's there are some big challenges there, but also the opportunities for us are also endless as well. Pharmaceuticals, I think we've obviously all lived through and still living through a global pandemic, and that's really just highlighted some of the ways that we need to work in the future. Looking at our manufacturing capacity, being able to respond and rapidly build up new and alternative manufacturing capacity and routes, rapid design and build solutions. I think if you think about the whole issue of providing the vaccine program and the testing program, we've had to respond much more quickly than we ever have before to this sort of issue. And I think that will be a theme going forward. As pharmaceutical engineering teams, we need to be much more responsive and reactive. And then thinking about things like targeted medicines for individual patient delivery, cell and gene therapy, etc. So again, lots of challenges, and much of that met by through biochemical routes. And finally, energy. So obviously a very big topic. I think in this case, there's no one size fits all. There are lots of different solutions which will contribute to the overall strategy going forward. But we're thinking about making sustainable energy, better use, reduced use of our fossil fuels, net zero, the impact on our environment, and again, many biochemical routes in as part of this solution. The next generation of alternative fuels and renewable energy sources, looking at how we can use organic routes to produce energy. So again, lots of opportunity for us, lots of challenges to address. And then finally, the last point really is about thinking about how responsibly we use the energy that we're provided with. And I think that's something that WSP as an organisation, as well as helping our clients to better use their energy and to reduce their own carbon footprint, we're also looking internally and to provide a future for us which reduces the amount of carbon that we use in delivering our designs and advice and consulting and projects. So that was a whistle-stop tour to biochemical engineering as I see it, and WSP and some of the work that we're involved in. And I think now it's time to move on to the main event of this morning, which is hearing a bit more about some of the fantastic projects that we've been working on as part of our global IChemE award process. So I'm happy to take questions, but I think we probably want to move on to the main event. Thank you very much for listening.
Okay, thank you very much, Simon, and thank you once again to WSP for its kind support of the IChemE Global Awards. So our awards judging panel has been hard at work in recent weeks to review and rank all of the entries, and these are our finalists. We have Clean TeQ Water, Micropore Technologies, a joint entry from the National Institute for Innovation in Manufacturing Biopharmaceuticals and the Bioforum Operations Group, ProClean Technologies, the University of Malaya, and a joint entry from the University of Nottingham, Malaysia, Algae Sites and Biolina Cooperation. 4 of our finalists join us today to tell us more about their work and answer your questions. You can get in touch with us using the questions box at any stage during today's webinar. You should see the questions box on screen. So let's get started with our first presentation. Representing Clean TeQ Water, it's Jonathan Wright.
Yeah, hi everyone. If you can see my screen, okay?
Yes, we can, yes.
Great. So yeah, thanks for the introduction and welcome to our webinar. We're really honoured to have been shortlisted as finalists, both in the Biochemical Award and the Innovative Product Award yesterday. So getting a second bite of the cherry today, going to introduce again our encapsulated bacteria technology that's been in development over the past year or so. Just to set the scene, elevated nitrate levels are a major issue around the world. Have you lost my screen there? Sorry, can you still see that?
Yes, we can, yep.
Yep. Yeah, elevated nitrate levels are a major issue around the world. Issues often arise from municipal water treatment as well as mining and industrial wastewaters. Discharging those nitrate contaminated wastewaters into natural water bodies can be a key cause of eutrophication. That's a contributing factor to algal blooms, and that can lead to major fish kill events, and that impacts obviously waterways and the communities that depend on those healthy aquacultures. So many countries, including China, are introducing stricter nutrient discharge limits on nitrogen and phosphorus, which is intended to limit that environmental impact. Potable water is another sector which is negatively impacted by nitrate pollution. Groundwater is contaminated with nitrates, a major global health issue, and that can lead to conditions such as blue baby syndrome and increasingly being linked to forms of cancer. Conventional bacterial treatment methods that you might find in wastewater treatment using free cell bacteria, such as CAS, MBR, MBBRs, and BAF, these can struggle to reach new limits of typically set less than 1 ppm these days for total nitrogen, while RO and batch ion exchange processes can remove nitrates as well, but they do produce large volumes of concentrated waste that needs to be disposed of. So just to introduce our technologies which have been developed, firstly the BioCleanse, which is a biological technology in which microorganisms are encapsulated in a PVA gel carrier. This maximizes the diffusion rate and also protects the bacteria from saline and harsh conditions, which we've found enables consistently high nitrification and denitrification activity to occur. We've coupled this with another technology that we have in-house, which is called continuous ion exchange, or CIF. And the coupling of this is now what we have as a patent pending system called BioNex. In this approach, firstly, we use continuous ion exchange to concentrate nitrate from low levels that you find in the wastewater up into very high concentrations, where they're then fed to the BioCleanse, which operate at very high activity rates, completely removing the nitrate from the system and producing inert nitrogen gas into the atmosphere. So this has a number of benefits, including very high recovery levels, low amounts of waste, and very high reductions in chemical consumption. So if we take a look at what the BioCleanse look like, the encapsulated bacteria, which are selected either for nitrate or ammonia removal, these are held within a porous matrix that the substrates can pass easily through. And this helps to keep the bacteria actually within the cells themselves. So rather than free cell floating around a reactor, the bacteria all live inside this PVA gel carrier, and the substrates pass to and from this matrix. And the unique aspect of this technology is the ability to maintain very high efficiency levels in very saline conditions at or above that of seawater, or in very high strength brines, which are typically used for ion exchange regeneration, where standard free cell bacteria cannot easily function. Now here's a few close-ups of what it looks like under SEM imaging. So you can see the porous permeable sponge-like structure, which is carefully controlled during manufacturing processes to provide a very suitable space for these biological communities to grow and thrive. So we've started manufacturing these on a large scale in the last year at a facility in China, currently with a capacity of around 40 tons per year. And we've already got a number of users in that region being used across aquaculture and wastewater treatment. So we take a short dive into what denitrification is. This is the process of converting nitrate to nitrogen gas through bacterial technology. With the BioCleanse bacteria, this occurs when oxygen levels are low, so under anoxic conditions, typically when the dissolved oxygen is less than about half a ppm. And then when the oxygen levels are depleted, nitrate becomes the primary substrate. When bacteria break apart the nitrate, it's first reduced into nitrite, NO2, and in turn into nitrogen gas. A carbon source or organic substrate is required for the reaction to occur. Typically, we use acetate or methanol or ethanol. And if there are simple chain organics present in the water already, these can also be used as a food source. In the Bionex approach, which has been developed over the last year or so, we employ continuous ion exchange, as mentioned, to concentrate the nitrate. So in this example, we've taken it from about 5 to 10 ppm up to around 200 ppm. And the treated water coming out of the ion exchange process can be controlled to less than 1 ppm, which is suitable for low levels of discharge. So the limiting factor with typical batch ion exchange processes is that they can be affected by bacteria and sludge and causing fouling in the system. But the unique aspect of our countercurrent ion exchange process is that we are continuously moving resin counterclockwise to the water going through the system. So we can actually manage levels of fouling very easily. The treated brine is then filtered after coming through the BioCleanse, where we purge typically less than 1% of the flow of the whole system. The waste from this filtration process can be returned upstream if there's an existing wastewater treatment plant, or it can be blended with the product or back into the feed, so we can maintain a zero liquid discharge approach, which is very important these days in terms of reducing footprint of evaporation systems. Salt usage is also minimized since the majority of the brine is reused in every regeneration cycle. And this is again a downfall of batch processes where this concentrated brine with the nitrate isn't being treated and is being released into waterways. Research and development has resulted in the use of using ultra-high specific nitrate resins for this process, and that's allowed us to manage feed waters that contain significant competing anions, such as chloride and sulfate. So in the case that there is a high sulfate to nitrate ratio in the feed water, which is often the case in when you're trying to treat tertiary wastewaters, the system can actually be tuned here to provide a unique desorption step without adding any additional infrastructure or reagents being used, and we can consistently achieve a low level of nitrate coming out of the end product. So a full-scale plant is typically what you might see here. You can see on the left-hand side the continuous ion exchange system, which has a number of columns for adsorption, desorption, and wash. The BioCleanse reactors typically have 1 or 2 slowly agitated reactors with a special mixing blade design installed in them, which is low shear. Other components of the plant typically include some process tanks, dosing systems, and electrical and control systems for full automation. We actually do have a full-scale flagship project, which is due to be commissioned at the end of the year. This is for 12 megalitres per day, which is going to treat coal mine wastewater up in northern China. We've also been doing a number of pilot trials for customers. In Tianjin in China, we've been testing for around 6 months to provide a proof of concept to a client who's struggling to reach a low set of discharge limits with their existing system and currently being fined for it. So our pilot treated up to about 100 cubes per day with a skid-mounted CIF system, ion exchange system on the left here, and 2 stages of bioreactors. You can see the inside of the bioreactor on the right in the video. The lenses that do look brown, that is not because of sludge, but it's because of how they actually turn when they're operating at high levels of activity. So they come off the manufacturing process clear, and then when they're operating at high levels, they get browner and browner in color. We use a proprietary self-cleaning screen as well. It's been developed to keep the lenses within the reactor, allowing the wastewater to pass through. Just to have a look at some of the pilot results, this is what the Bionex overall looked like over quite a long period of time. We reduced from about 20 ppm of nitrate to basically less than 1, which was more or less undetectable levels. This resulted in a brine coming off the desorption step of about 200 ppm. So then looking at the bioreactor themselves, after a few days of the bacteria warming up from cold storage, which is typically what you find for bacteria, they need some time to wake up. Then the 2 stages of reactors were found to consistently remove nitrate down to less than 10 ppm. So this green curve here is what happened coming out of the second reactor. We were about halfway through, we decided to try and reduce the carbon to nitrate dosing, which we did a bit too much to start with, and then eventually managed to optimize it and find a level that was optimal. We were also, in a number of other trials, we also managed to reduce the amount of loading of bioclens that we had in the reactors. So here we had about 10% loading of master volume. We've actually found that we can get down to as low as 2% as well, with activity rates around 1,000 or even higher in terms of milligrams of nitrate per kilogram of lenses installed. And this is all with only about 2 hours of residence time in each of the reactors. So just to conclude, we believe that the encapsulated bacterial technology, coupled with our innovative continuous ion exchange process, can provide a fundamental shift in how we apply bacterial solutions to water treatment in reaching very low surface discharge levels for the future. And we believe this is going to provide a route to enabling reuse of wastewater, as well as reducing demands of clean drinking water for other purposes. And obviously that can help to reduce harmful algal blooms and eutrophication of water bodies through reducing those nutrient loads as well. The resulting process has very low OPEX. We typically use less than 10% of salt that a typical batch process would use, and the cost of treatment is typically less than 10 cents per cube treated. And we're not stopping there either. We're looking at future developments, including phosphate removal as well, trying to recover a solid reusable phosphorus product through using specialty doped ion exchange. So that concludes our presentation. I hope you've enjoyed it, and we'd like to wish all the finalists best of luck.
Okay, thank you very much, Jonathan. We'll pause for questions now. So if you would like to know more about Jonathan's presentation, please get in touch through the questions box. We do have our first question. How often do we reduce the bacteria loading? Or in other words, how do we remove the bacteria that's grown in the system?
So the bacteria basically stay inside the cells. The colonies do grow up to a certain level when they get to high activity rates, and there's a bit of shedding if the activity rates do change. But otherwise, there's no need to do any sort of maintenance in terms of removing or adding bacterial cells. We carefully select what we need to install initially, and there's always the opportunity to increase capacity by adding more cells to a certain reactor if required over time as well. But generally, yeah, there's not a lot of maintenance you need to do with these encapsulated bacteria compared to using free cell bacteria, which you may typically expect to have to do a bit more maintenance on that side of things.
Okay, thank you. We're going to move on now. So thanks for getting things started, Jonathan. It's time to move to our second finalist. Next up, representing the joint entry, or the entry from Micropore Technologies, I should say, it's Di Hayward.
Hello, good morning. And let me... So, good morning. We talk about, we had the introduction from Simon Lewis, where he mentioned vaccines and the way they've changed the world over the last 18 months or so. Micropore Technologies' entry in this category is around engineering the design for manufacture of vaccines of the mRNA type, which use lipid nanoparticles, LNPs, as an integral part of the way that they are manufactured. LNPs are integral because they're needed to be able, if you like, to smuggle the RNA into the body without it itself being recognized as an alien invader. So traditionally, micromixing is the key to manufacturing good quality product, and typical manufacturing routes have historically used either microfluidics or impinging jet stroke T-junction mixers. The scale at which these operate is not continuous in as much as microfluidics run at the small scale but do not scale up to the sort of scale and quantities that Pfizer and Moderna are using. So they use a different technology, which is impinging jet. And there are various pluses and minuses around those, but what we set out to do was to design something that would seamlessly go from the smallest quantity that you might want. And because materials at the early stages, well, all the way through, are rare and expensive, mRNA is extraordinarily expensive, you want to start with a very, very small quantity, but you want to be able to build that up consistently and reliably on the basis of good scalability. So on the left-hand side, we've got what the market told us it wanted, and on the right-hand side is our response to that, which is the ability to produce the AXF Mini, which is in dimensions, it's about 2 centimeters long and about, without the clamps, and about 1.5 centimeters in diameter. It's very easy to use, and it's a very, very small volume. And if we go on up then through to the next stage, on the basis that you can use that for very early experiments and lab experiments, we'll see that it's very flexible to be able to go on to clinical materials, because if you run it for longer, you are capable of creating 20 liters per hour of product, which is big enough for certainly clinical trials, and in fact, even some small patient populations. So you've got something that's really very small, very flexible, and allows you good batch repeatability. And because it's all stainless steel, 316L stainless steel, electropolished, it's capable of being, it is GMP compliant, and we provide all the necessary documentation and support for that. And then into manufacturing, if you really do need the sort of quantity that is there for Pfizer and Moderna, the AXF1 shown on the screen here gives you up to 200 litres an hour and has still the same issues of no moving parts, no consumables, very easy operation, and both have the capability of doing a process analytics control loop, which means that in real time you can measure online what's going on and correct for any deviations by altering the process parameters. And I'll come to those in a second. But it's a quality by design approach to this. And as I say, it's an integrated system that would give you, if you wanted to, straight through flow into a fill finish in a vial. What are the results? Well, one of the nice things is that it's very tunable. Change the process conditions and you can change the size of the lipid nanoparticle containing your RNA at will in a very predictable way. So here we are, we're showing that we can do this at 120 mils a minute, produce material that's around about 100 nanometers. And if we increase the flow rate, we can come down to about 60 nanometers. And on the left-hand side is a very typical formulation for a model, well-recognized formulation. We also have control over the components involved in the device, such that if we change the pore size of the internal membrane or the dimensions of the annulus, then again we can control in a very predictable way. We can manufacture product to precisely the specification that our clients want. And that's really rather good. That's very useful because it gives a lot of flexibility. And as I said, the fact that we can predict it means that you can include that PAT loop. All very well, but if it doesn't carry enough RNA, then it's not very good. You'll see here encapsulation efficiency of 98% of the RNA, which is higher than most other ways of doing it. In fact, it's industry leading quality in terms of the product. And the biological results are that it's just exactly what you would expect. No toxicity in an in vitro study, concentration dependent uptake, which again is what you'd expect, and a good dose response curve on the right hand side. One of the key things is that this is a very low shear process. And as I've mentioned earlier, RNA is expensive, but it's also very fragile. And one of the things that we can demonstrate, as shown here, is that before and after our process, using the AXF Mini, we have no degradation of mRNA in the process. That compares and contrasts with the other processes, which are high shear and high energy processes, which are damaging to mRNA. So from all perspectives, there's a win-win-win all the way around on this. And just to illustrate how simple this is, because I set up the design criteria with our engineers to say, I want this to be a very simple thing to operate. So I had my grandson in, who came in for a day of work experience, and he had no familiarity with any of this sort of equipment before. So he set up the device, he presses 2 syringe pumps, the buttons on 2 syringe pumps to get them pumping. And we can see the material flowing through into the AXF Mini and then on to into the sample collection beaker. And at a point, you will see him change over. There we go. Change over and start collecting a different sample. And if you change the flow rate, you can start collecting a different sample again. And you can do that for as many samples as you like while you still have a syringe full. So you've been able to produce initial material but that same device will go up to full-scale manufacturing at 20 litres per hour. Ladies and gentlemen, thank you very much indeed. And I'm now happy to hand back to the controller, the host, and take any questions.
Okay, thank you very much, Di. And in all my years working on the IChemE Awards, I think it's the first entry to involve the grandchildren, so extra bonus points for that. Once again, we'll pause for Q&A. So if you haven't already and you would like to get in touch, now's the time to do so through the questions box. And while I give you a moment on that, don't forget there are more IChemE Awards coming up, IChemE Award webinars coming up next week. You can find out all the dates and times by visiting our website, icheme.org/global awards. We don't appear to have any questions, so we're going to move on. Thank you very much, Di. We'll move on to our third finalist of the day from the NII MBL and Bioforum Operations Group. Please welcome Kevin Gibson and Russell Jones.
Yeah, good morning, everyone. It's really good to be here as part of this session this morning. I'm Russell Jones. I'm going to be presenting today with Kevin, the work that's been done with this collaboration with Nimble and the Bioforum Operations Group, where we've really developed and looked at improving the buffer preparation, which is a challenge today. So I don't expect you to read all this, but essentially the preparation of and storage of buffer solutions during the manufacture of therapeutics is an essential function that drives plant schedule, capacity. It requires dedicated and significant footprint, labor needs, and cost. So it does have a significant impact on the cost of manufacturing and can result in process and facility bottlenecks, which in turn lead to reduced overall productivity. So the Bioforum technology roadmap had identified this as a critical need and formed a buffer prep work stream to identify solutions. Further on, then, we worked on a collaboration with the National Institute for Innovation in Biopharmaceutical Manufacturing, so Nimble, to design and build a fully scaled cGMP-ready system that was designed to deliver buffer on demand. So last year, the system was constructed, installed, and extensively tested at the facilities of Nimble in the University of Delaware with a team from both Nimble and Bioforum. And what we're going to do is share the outcomes of this work in this presentation. And just for reference, an article has been submitted for peer review publication, and the design and data sheet from the work that's been done is going to be made available open source to encourage adoption of this development, the buffer stock blending system. Next slide, please, Kevin. So if we look at the buffer management problem, essentially upstream intensification and process optimization is driving our industry towards smaller and more flexible operating footprints, biomanufacturing facilities. Now, many of these are going to be comprised primarily of single-use or hybrid technologies, and these facilities are capable of producing a significant amount of drug product. Knowing that downstream operations are product mass-based, any increase upstream, any increase in productivity upstream will lead to a proportional increase downstream for the buffer demand. And when we look at the numbers here in this slide, which is a real example from one of our team members, we can clearly see the problem. So in this case, the facility here is producing a ton of bulk drug substance per year. It requires a significant amount of cell culture media and an even greater volume of downstream buffer. So we're looking at 17 unique buffers, and to manufacture those, there's more than 500 batches needed to be made per year. So essentially, buffer prep as a process has not really changed and has not kept up with the upstream developments in the industry that is driving towards smaller, more flexible operating footprints. And traditional buffer prep and supply is going to become a bottleneck, hence the need to look at these alternative ways of manufacturing and delivering buffer to a process. Next slide, Kevin, please. So really, this collaboration of consortia has worked hand in hand to get to where we are today. And it started with the Bioforum Operations Group, BPOG, And there's a quote here from one of our members: the facility, Natalie Frau, the facility of the future will not be possible if we don't find a solution for the buffer prep bottleneck. So BPOG essentially forms sub-teams to address the gaps, to figure out what the problem is and how might we close these gaps. And the team, were broken into chemistry, chromatography teams, modeling teams, engineering and automation teams. And the design concept that was put forward was essentially, when you make a buffer, there are 4 components: an acid, base, and a salt, and then water. So these 4 components are sufficient for the majority of buffers, and you could have 6, maybe 8 stock solutions, and the blending and mixing and diluting of those stock solutions could deliver upwards of 15 buffers downstream. So it really was a simplified approach. We then reached out to Nimble, and a proposal was made by Merck & Co, one of our members, for Nimble to co-fund the project. We developed the proposal then to fund, build, and test the buffer stock blending skid, and it was 80% funded by Nimble, 20% BPOG members involved. And all that work was done last year, as I said, in the University of Delaware, with teams from the companies listed here: Merck & Co, Merck, Janssen, Sanofi, GSK, Cytiva, and Takeda. Next slide, please, Kevin. So really, why are we doing this? Why did we take this approach? We wanted to democratize technology, make it available to all, and to come up with a solution that would really help simplify buffer manufacturing and production, really reducing the cost of that process and making it more value add rather than non-value add. The work that we've done last year is really to generate all the data to demonstrate that this concept does really work, because in doing so, we really believe we can accelerate adoption within the industry of moving buffer prep from the old-fashioned ways of doing things to something far more suited to the manufacturing requirements that we have today. Kevin, over to you.
So on the screen board, you can see in the top, that's the skid that we built, and it's installed at the University of Delaware at the Nimble headquarters. So the key things with the design, it's a fully GMP design, and we've simplified it, so it's mass flow based recipe control. And the key advantage of the system is it's highly flexible. You can connect it to single-use bags, to tanks. You can go directly to your chromatography skid. And as Russ mentioned, it's an open source design. So you can take this design and build on it for future systems. And the idea of the project is to really drive change in the industry. The skid we built is sized for a 2,000 litre single-use bioreactor process, looking at 0.6 to 1.2 metre chromatography volumes. And we can make buffer at 8 to 60 litres a minute. It's highly flexible, where we've got 4 stock pumps where you can connect up to 16 stock solutions. The idea being you can connect all your stocks, do your setup, and make the wide range of buffers needed for the process. In terms of our achievements to date, so it's been a fantastic collaboration, and there's been great engagement across the different companies and organizations involved. Obviously, we've designed the system, we've built the system, and we've completed testing. To date, there's 2 white papers after being published. They're available on the Bioforum website, and there will be an addendum and a peer review paper coming soon. I suppose it's been a great testament to the collaboration of the team, how far we've come. We started off as a concept, and it's really the whole industry coming together to solve a common problem, and then sharing that design and making it open source and freely available. Those are the initial impressions and how the skid works. So the speed which you can make buffer is an absolute game changer. So when you're making buffer at 60 litres a minute, so if we compare to the traditional buffer prep, it might be 1 prep per tank per shift. So you need an entire shift to make, say, 3,000 litres of buffer in a tank. With this system, you make that same amount of buffer in less than an hour. And you can see on the left, when we're making buffer within spec, within pH and conductivity requirements. Also, it's a bit different in that you do have some stock solution going to drain. But when we've done the assessments, the efficiency is 95 to 99%. So your overall wastage from buffer actually decreases significantly compared to traditional buffer prep. Looking at the benefits, I suppose the idea is that this system can have a significant or transformative effect on facility design and operation. So by making buffers from a common set of stock solutions, to make 15-20 buffers, you might need, say, for example, 7 single component stock solutions. So now you're down to making 7 solutions per batch instead of 15 or 20. The fact they're highly concentrated single component, now you don't even have to make them every batch. You can keep the volumes relatively small and still make them for multiple batches, or even have the opportunity to outsource. Also, through significantly reducing the volume of liquid to be stored and handled, in the case of going direct to a chromatography process, you can eliminate the need for buffer hold completely. I think we did a detailed economic study at both 2,000 litre single-use scale and 12,500 litre scale. We looked at that for a wide range of product titers from 2 to 10 grams per litre in both cases. In every scenario, the use of buffer stock blending reduced the overall net present cost and the overall cost of buffer. And the main savings are on the operational cost. So at the smaller scale, because you're making buffers so quickly, it's a lot less labor intensive. So there's significant savings there. And also you reduced consumables costs through the reduction in volumes. At the large scale, even the capital cost goes a bit lower. So it's savings all around, with the exception of consumables costs. And while that's shown as a red, it's really a positive. Because of you're allowing much greater utilization of single use at the large scale, which gives a lot more flexibility, and also there's environmental benefits. So there's been a lot of life cycle assessments done showing that single use is superior to your traditional stainless steel systems, and that's down to the large volumes of WFI needed for cleaning. In this case, we're allowing greater utilization of single use, and even at a single use scale, by reducing the volumes, we're reducing the absolute amount of single use components needed. So in conclusion, the traditional buffer prep requires significant amounts of labour, capital, footprint and time. Typically, it can account for about 20% of the overall cost of manufacturing, looking at your capital and operating costs. The Bioforum looked at identified the potential for addressing this area in the technology roadmap and identified it as a key problem that's common across the industry. Through Bioforum, we put forward the initial design concept, and then that design was funded through Nimble, and we've built and performed testing on the skid, basically showing that the concept, it really does work. And we can produce a very wide range of buffers in real time and go direct to the process. And the whole open source nature demonstrates a technological advance with the potential to transform the industry. We like to think this project can inspire other similar projects where the industry is coming together to solve common problems. There's no one company on their own has been able to solve this problem. And it's a high cost, it's creating bottlenecks in industry. And by coming together, we're hoping to change that. So the last thing to mention is, when I mentioned a lot of the industry coming together, you can see on this slide how many companies and individuals have come together to make this project happen. And again, I'm not going to go through them all because with the amount there, I'll no doubt forget somebody. But it really is the entire industry coming together. Happy to take any questions. Okay, thank you very much, Kevin and Russell. So we'll pause once again for questions. If you'd like to get in touch, please use the questions box now. And while we just pause for a moment on that, if you have been suitably inspired by what you've seen today, it might be time to start thinking about your own awards entry next year. Keep an eye on the IChemE website for more details, but we plan to open for entries in early March 2022. Okay, we don't appear to have any questions, so thank you, Kevin and Russell. We have reached our final presentation of the day, Representing Clean Technologies, it's Shivaram Pillai.
Thank you so much for the opportunity. It's an honor to be the finalist of this Biochemical Engineering Award. I present this on behalf of my team here. We are a company based here in India, in southern part of India, and our main business is in replacing chemical surfactants and some other kinds of chemicals used in processing industries with a biodegradable non-toxic option made using a probiotic technology. I want to spend a minute or so on talking about how we do things. If you compare the traditional fermentation-based manufacturing processes, which is shown in the upper part of this slide, you start with a single species of microorganism, which is efficient in producing any compound of interest, subject it through a fermentation system in a bioreactor under controlled temperature and aseptic conditions. And at the end of the fermentation, take the broth and pass it through a downstream processing step to finally come out with the compound of interest. And then subsequently, that compound is formulated in the end-use product. Literature shows, and my own experience shows, that 60 to 80% of the total production cost is actually in the downstream processing steps. Compare that to what we actually do. We have some unique approaches to making our product and significantly reducing the cost. Number one is we use a consortium of microorganisms in the culture which we use. The culture we use typically contains about 15 different species of microorganisms. And using locally available feedstock, we carry out the fermentation under some unique conditions. One is that we don't heat or cool the fermenter. The fermentation happens under ambient temperature. And because we have a unique consortium, which is very sturdy, we don't need to have aseptic conditions. And so we have very simple vessels on which we conduct the fermentation process. So overall, the capital cost of the facility is a fraction of a typical fermentation-based industry. And finally, the other unique feature of our process is at the end of the fermentation, the end-use product, which is to be used by the customer, is ready. And therefore, we don't have to go through a downstream process step because the broth at the end of the process does not contain any unwanted compounds. So essentially, we are reducing the cost of production by more than 60–65% by simply eliminating the downstream step. And then by not using any energy during the production, we are further reducing the cost of production. So this is what helps us to be very green in our manufacturing process as well as low cost in our manufacturing process. So the products we make are readily biodegradable. They're cost competitive because of what I mentioned, and because the microbes we use are all GRAS listed microbes, it is extremely safe for the people who are manufacturing the product. Low energy, very low waste generation, simple, low cost fermenters, highly safe for the workers, no downstream processing. Coming to the application for which we filed our application in the Biochemical Engineering Award section is a approach to reducing or eliminating odor in pulp and paper mills. Pulp and paper mills have a major challenge in terms of odor. Those who work in the industry know very well this challenge. Current solutions for this problem are aimed at killing the bacteria using biocides, including hypochlorite, so that the bacteria don't produce, or the bacteria which are responsible for producing the volatile odor compounds are no longer surviving in that process conditions. While they get reasonably okay results, it is not completely satisfactory to the customer. That is where we come in. We have developed a first of its kind formulation using the proprietary probiotic platform, which I described a little while ago. This whole system eliminates use of biocides and hypo. So we significantly reduce the amount of chemicals which go into the operation. Manufacturing, of course, is low carbon footprint, and I talked about the way we manufacture is a circular process, and it addresses the root cause of the odor problem, which is the bacteria which are responsible for producing the volatile fatty acids. The product we use is a combination of probiotic microbial consortium along with biosurfactants produced by these microbes. So it's a product which contains live probiotic bacteria as well as biosurfactants. The mechanism is very simple. The biosurfactant which is present in the product breaks up the existing biofilm of the pathogens or the bacteria, anaerobic bacteria which produce these volatile odor-causing compounds. Once you break up the existing biofilm, the probiotic bacteria which are present in the culture which we apply, they begin to establish beneficial biofilm on the surface of the vessels or the pipes which are in the process conditions. And once the probiotic blanket is formed, pathogenic bacteria find it impossible to establish a biofilm again. Thereby, they don't survive in the system, and hence no volatile fatty acids are produced. So very simple approach, however effective in producing the results which we are looking for. So paper mills normally have two main objectives when they are looking at the odor control program. One is elimination of the ambient odor in the process area. This is typically measured by the ORP levels in the pulp which they process. So they measure ORP at various points and record. If the ORP is maintained in a positive range, let's say between -25 and above, then they know that the odor causing bacteria will not thrive in those conditions, and therefore there will not be any odor. The second objective the mills have is not to have any odor in the final paper which is produced by them. Now this they track by measuring the volatile fatty acid content after the finished paper is made. Lower the concentration of VFA, the better it is, and it indicates that there will be no perceived odor in the paper. So once we started to use our product as against the biocides and hypochlorite they were using, we started to see that ORP improved. It came into the positive range compared to when it was negative before. Odor in the process areas within the mill, the operators of the mill perceived that there was no longer any odor, made the place better to work in. The effluent treatment plant showed water with clear water within a month of us starting operation. Of course, biocides and hypo was completely eliminated, and in the finished paper, we saw more than 30% reduction in VFA. This slide actually shows how the ORP numbers moved in the pulp which was being processed. Before we started applying our product, when hypo and biocides were used, it was still fluctuating and typically in the negative region. And within a short period of starting the application program with our product, it started to build and become positive. And over a period of a month or so, it stabilized in very positive range and continued to be maintained at level. And after a couple of months, the operating mill just wanted to check if they shift over to biocides and hypo again, whether they would be able to get the results which they were getting with our product. And so they stopped dosing our program. And again, we saw that the ORP levels started to go down into the negative region. They restarted with our program and the ORP levels came back into the positive numbers. So clearly, the approach we had taken serving the purpose and giving them the results. The finished paper volatile fatty acids were analyzed, and over a period of time after they started our program, there was a significant reduction up to almost more than 50% reduction in the VFA to a level that there was no longer any perceived odor in the finished paper. So we were able to achieve both the objectives of the mill with this very simple approach which we took. A picture of the mill, what you see on the top are pictures of the inside of the mill where before they started on with our program where they were using biocides and hypo. you can see significant levels of biofilm and slime being formed in the pipes and the screens. After 3 months of starting with our program, very clean system seen with hardly any growth of slime or microorganisms. Proclean as a company has been working over the last 7-8 years, and over this period of time, we have stopped 7,200 metric tons of carbon dioxide from being released into the atmosphere because we replaced chemicals which were using, customers were using before. And in the process of having a green manufacturing system and a biodegradable product, we have reduced this carbon footprint over the last few years. This is almost equivalent to something like 6.3 million passenger kilometers of an aeroplane. By replacing the chemicals, we have also stopped over 11,000 metric tons of chemicals being put into the atmosphere for process conditions. A view of our production facility, very low cost vessels being used in ambient conditions to manufacture our product. That's it from me. Thank you for this opportunity to present to you, gentlemen.
Okay, thank you very much. So for the last time today, we'll take a pause for Q&A. If you have a question and you want to get in touch, please do so now through the questions box. And while I give you a moment on that, don't forget the winner of this award will have a chance to lift a second prize before the end of the month. All of our team award winners qualify as finalists for our top prize, the Outstanding Achievement in Chemical and Process Engineering Award. We'll be announcing the winner of that prize next Friday, Friday the 15th of October. Okay, so we have some questions. Does growth of consortium vary because of the temperature?
The microbes in the consortium, especially for responsible for dealing with the odor crossing bacterium, are stable at high temperatures. In fact, they are purple non-sulfur microorganisms, so they can work at temperatures up to 125 degrees Celsius.
Okay, thank you. Next question: are there any other applications for this apart from paper plants?
Yes, we do. We do make products for textile processing industry. We also look at water treatment applications. So places where surfactants need to be used, we have applications using this technology approach. Typically, the products containing the live microbes are used wherever there is BOD or COD to be reduced. While the places where such requirements are not there, only the biosurfactants are used. We have multiple applications like degreasing or soaping or washing away.
Okay, thank you very much. We're going to leave it there and move on because it's time to find out who the winner of the IChemE Biochemical Engineering Award is. Before we do that, I'd like to thank all 6 of our finalists, and particularly the 4 that have been able to join us today to tell us more about their work. I'm sure you'll agree, a fine, diverse range of work on show today. So let's start with our highly commended entries, and they are micropore technologies and the joint entry from the National Institute for Innovation in Manufacturing Biopharmaceuticals and the Bioforum Operations Group. So well done to everybody associated with those projects. But now, to announce the award winner, I'd like to welcome back WSP's Simon Lewis.
Thank you, Matt. Ladies and gentlemen, the winner of the IChemE Biochemical Engineering Award, sponsored by WSP, is Clean TeQ Water. Many congratulations.
Thank you, Simon, and congratulations to Clean TeQ Water and everyone who's worked on our winning entry. Jonathan, I hope you're still with us. If you are, tell us, how does it feel to be an IChemE Award winner?
Yes, thank you very much. It's a great honour to be named the winner of this prestigious award. So I'd like to thank everyone in the team across both Australia and our team in China, who's put a lot of work and effort into developing the technology of the last year or so. So yeah, well, very grateful indeed, and obviously a lot of great entries have been put into this category this year. I think they're all worthy winners, so we're very, very honoured to have been recipients of this fine award. So thank you very much.
Well, thank you for entering and congratulations once again. A big thank you to everyone for joining us today, particularly our presenters and of course our sponsor WSP. We've still got plenty more IChemE Award webinars to come next week, and we'll be back this afternoon to announce the winner of our Research Project Award. But that's all for now, so thank you very much for joining us.
Thank you.